So dear students, so today our topic is glycogenesis, that is glycogen synthesis. Okay, it is a process, it is exist in our body, so where the excess of glucose, so after utilization, so that was stored in the form of the glycogens. Okay, so glycogen nothing but it is our FD for carbohydrates. So excess of glucose, so that was stored in the various cells of the liver as well as the in the muscles, and some stresses it was stored in the kidney. Okay, so after daily utilizations of glucose, so that excess glucose is stored so in the form of the certain biomolecule, so that biomolecule is the glycogens. Okay, glycogen nothing but it is a polymers. Okay, so of the carbohydrates, so that process is involved in the synthesis of the glycogen, so that process was known as the glycogenesis. That is the genesis of the glycogen, that is the excess of the carbohydrates. So regarding the glycogenesis, it is the synthesis of glycogen is known as a glycogenesis process. It is one of the important process or pathway, so that was helpful for also to manage the sugar level in body, as well as in the case of the deficiency it can supply the glucose to the our body. Okay, for shorter durations. Okay, so glycogen is mainly stored in the liver as well as in the muscles. So dear students, the 60% of the glycogen, the sorry 60 not 6%, the weight of the liver, so that is the where the glycogen can be stored, about 6% weight of the liver, and as well as 1% muscle weight. So that much of glycogen is stored in our body. For example, the liver weight and average is 1.5 kg. Okay, it is about 1.5 kg. It means 1500 gram. So 6% of liver weight, so the glycogen is around 90 gram. So that is 80 to 90 gram of the glycogen, so that was stored in the liver. But in our body the muscles are more, the percentage is less, that is only 1%, but the muscle mass in our body is more. So that's why the glycogen is stored in more in muscle, that is 3 to 4 times more glycogen stored in muscle as compared to liver. Only one reason, the muscle mass is more than the liver weight. For example, and average, the healthy human beings, so they will be having the muscle mass around and average, okay, 30 to 35 kg. Okay, so here once again the muscles is more present in the woman as compared to the male. So this 30 to 35, it is an adult weight. Okay, so that way they will be having the weight around, that is a 65, that is 55 to 70. Okay, so those people and average, so they will be having the muscle mass, that is a 30 to 35 kg, and 1% of muscle weight, the glycogen was stored in the muscles. So that's why, so in the muscle the glycogen is stored around 300 to 350 gram. So like that, liver only have 90 gram, and the muscle and average it will be having the 300 to 350 grams. So that's why the muscle contain more glycogen, so as compared to the liver. And the total is, that is the 390 to 440 gram of glycogen or FD is present in our body, that is around and average. So glycogen in our body, so that should be 440 gram plus or minus maybe 40. So that much glycogen or our FD will be having in the case of the starvations. So already you know, in the resting positions, the body is required 160 gram of glycogen in resting. Okay, there is no work. So only resting conditions. So our body, it will be required 160 gram. If the body, if we are involved in the exercise or daily regular activity or mental activity, especially official works or intelligence work, so where the intake of glucose is more. So that's why already in the yesterday class I told to you, so the glycogen storage is for shorter durations. Okay, within 24 to 36 hours our FD was finished in the case of starvations. So in the case of the hunger, in the case of the hypoglycemia, the glycogen can supply the glucose only for shorter duration, because it is in our body, it is only present the 440 gram and average, and in resting position we required 160 gram. If you're involved in the work means it required more than 300 gram. It means within 24 hours our glycogen storage was finished. So that's why, so God made another arrangements for the source of glucose. So that process was known as the gluconeogenesis. Already we are discussing in the last class, that is the importance, and here we are discussing regarding the pathway of glycogenesis or synthesis of the glycogen. So that was taken place in the liver and muscle after the excess of glucose remain in the blood circulations. Okay, if it is not utilizes, okay, so up to how much, only it is having its own capacity, only around 440 gram of glycogen can stored. If still we having the excess of glucose in our food in our body means, so that glucose can counted into other compounds like a free fatty acids, or especially triglycerides are converted from the excess of glucose. So that's why even the pure vegetarian people, so they are prefer the more sweet means, so their food intake will be the more glucose, and utilization was less means, so only the 440 gram of the carbohydrates or glycogen stored, and remaining glucose, excess of glucose, so that was converted into a free fatty acids or triglycerides. So that's why the pure vegetarian people is also suffering from the heart attack due to blockage of the blood vessels or elevated of the free fatty acids, cholesterol or triglycerides, because in some cases the triglycerides are counted into VLDL, and triglycerides are also formed from the excess of glucose. Okay, then triglyceride come to VLDL, and VLDL is also responsible for in the liver, so they are interconnected LDL and cholesterol, and these all VLDL, LDL, cholesterol, triglycerides, so these all are considered as the bad fat. Okay, bad lipid. If their concentration increases the blood mean, so they can increase the load on the heart, blood become more and more thick. So to increase thickness of the blood, the pressure on the blood is increases. Then the person was suffering from the hypertension or high blood pressure. Due to that, the megaly is occurs. That is swelling of liver, swelling of muscle, swelling of heart, swelling of kidney, or swelling of lungs is taken place due to only when you are consuming the excess of glucose also. So that's why you should be have the limit on the glucose, if even if you are a pure vegetarian, because excess glucose on other side after finishing the FD of glycogen synthesis, then it is involved in synthesis of the free fatty acids and that is the triglycerides. So these are the bad fats. So dear students, so the glycogenesis, it mainly take place in the liver and as well as the muscle cells only after the excess of glucose, so that was remain in the our blood. Okay, and the glycogenesis is mainly stimulated hormone, that is insulin. So insulin, it can enhance the synthesis of glycogen in the muscle or as well as in the liver. So that's why the insulin was helpful for the diabetes mellitus or hyperglycemia, because it can enhance the synthesis of glycogen. So that process is helpful for to decrease the elevated sugar level in our plasma. So that's why the insulin is considered as the stimulator for the glycogenesis, and the glycogenesis mainly occurs in the liver and muscle cells. And the glycogenesis, it was inhibited by glucagon as well as the epinephrine. So epinephrine, it is the adrenal hormones secreted from the top of the kidney or cap of the kidney or adrenal gland. So that epinephrine, it can inhibit the glycogenesis, okay, and as well as glucagon. So it can inhibit the glycogenesis, because glucagon always releases when we are suffering from the hypoglycemia or low sugar found in the plasma or in the blood. Then only the glucagon was released from the alpha cells of the pancreatic gland. So that glucagon, it can enhance the glucose by inhibiting glycogenesis process, as well as it can enhance the breakdown of the glycogen. So like that, so these are three main hormones. So they are mainly involved in the regulations of the glycogenesis. Regarding the insulin, it can stimulate, and regarding the glucagon and epinephrine, so they are inhibit the glycogenesis. So now we go for the, so regarding the various steps, so those are involved in the synthesis of the glycogen. So mainly, so they are 6 to 7 steps. So they are involved in the conversions of glucose into a glycogen. So this process is mainly occurs in the liver as well as the muscle cells. Okay, so from the excess of glucose from the blood, okay, so that was enter into the cytosol. So in the cytosol, so in the presence of the hexokinase enzyme or glucokinase enzyme, it was counted into glucose 6 phosphate. Okay, so later on, so this the phosphoglucomutase is the another enzyme. It can replace the phosphate group on a glucose 6 phosphate and counted into a glucose 1 phosphate. Here one step here is the hidance. Okay, so one step here hidance. So it can be hided always, because so this phosphoglucose mutase first, it can add the phosphate group in glucose 6 phosphate at position 1. Okay, so later on it can remove the phosphate from the position 6 and remain phosphate in only in the position 1. So that's why it becomes the glucose 1,6 phosphate. So this step was the hidden only directly in many books. So they are mentioned the conversions of glucose 6 phosphate into glucose 1 phosphate. So beyond that, another reactions is there. So where this phosphoglucomutase first, it can add the phosphate group to the that is the one positions of this glucose or first positions of the glucose 6 phosphate, and later on it can remove the phosphate from the sixth position and it can yield the glucose 1 phosphate. So like that, so this phospho glucomutase, it is enzyme, it can convert glucose 6 phosphate into alpha, that is the glucose 1 phosphate. So later on, so in the presence of the UTP uridyl triphosphate and the enzyme udp-glucose phosphophosphorylase, so it is another important enzyme. So this enzyme, so in the presence of the UTP, so it can convert, that is a glucose 1 phosphate into UDP glucose or uracil diphosphate. So this is another key enzyme in the glycogenesis in the presence of that is a uridyl or uracil triphosphate. Okay, so that can convert, this enzyme can convert glucose 1 phosphate into uracil diphosphate glucose or UDP glucose. So it is the another steps of the glycogenesis. So later on, so this glucose molecule, so they are added on the that is alpha 1 4, Okay, alpha 1 4 glycoside bond. So one glucose lineup will be formed, so in the presence of the that is the glycogen synthase enzyme. So it can utilize the udp glucose and forms the bond between alpha 1 4 that is the glycosidic bond between the glucose molecule. And here, so this process, it can require the one molecule that is known as the glycogenins. So only, so this enzyme, it can add the glucose molecule on a glycogenin. So here the glycogen, it is the primer. So already primer is nothing but it is the composition, the molecule of the glycogen. Okay, it is the already existing glycogen. So where only, so this enzyme, when glucose is excess, it can add up. So where it can add up, so on the side of the non-reducing side. So the always the glycogen having two end, one is the reducing end and another is the non-reducing end. So reducing end in the having the only one end, but non-reducing it having the many residues. Okay, so this enzyme, it can add up the that is the glucose from the udp glucose on the glycogenin. And glycogen is nothing but it is a primer. Primer is nothing but glycogen molecule already exist in the liver cells, already exist in the muscle cells. So where it can build up the other molecule or add up the other molecule of the glucose. So that's what glycogen is nothing but it is one of the primer. That is the nothing but it is a consist of the glycogen or branches of the glucose containing molecule or primer that was known as the glycogenin. So, and this glycogen synthase enzyme, it is the another key enzyme. So this enzyme only active when the glycogenin primer is present. Without primer, so this enzyme not involved in the synthesis of the glycogen. So only the presence of the primer, so it can add up the glucose on non-reducing side by involving the formations of the alpha 1 4 glycoside bond. So like that, one by one the glucose molecule will be added up to 7 molecule. So after that, when the 7 molecule primer is ready along with the glycogenin, so this glycogen branching enzyme is active. It can create the bond between that is the glycogenin and 1 4 linkage primer. Okay, it can form the another bond, or that is the alpha 1 6 glycoside bond. So that was formed between the new chain and the glycogen primer. So like that, the glycogen molecule can be enlarged or new additions of glucose units can be added to the glycogen. In the presence of glycogen synthase, this was helpful for to add up the glucose from udp into the glycogen primer by alpha 1 4 utp, that is not utp, that alpha 1 4 glycoside bound with alpha. Then after that, so this glycogen branching enzyme, so when it, then this molecule is consist of 7 glucose. So then, so this enzyme, it can form the one link between, there is alpha 1 glycoside bound containing primer that of with the alpha 1 4. So between them it can forms the one bond. So like that, glycogen molecule will be formed. And here, so in the glycogen synthase, so from the glucose molecule, so this glucose molecule can release the udp, and it is the most important. So this udp is required for here as a utp. So when glycogen molecule forms, so this udp, it can reacts with the atp. So then it can forms the utp. So that utp is required here to convert glucose 1 phosphate into udp glucose. So that's where we require the utp. So that utp is supplied from the this reaction during the glycogen branching, there is a branching reaction. So where the released udp, it can react with the atp, form the utp. So like that, the glycogenesis, it can require the atp molecule. So like that, the glycogen synthesis or glycogenesis pathway, it can regulated in the 6 steps, and the two steps are the hidden. One is the here, so where the formations of glucose 1,6 phosphate diphosphate, and another one is here, that is the UDP. It can react with ATP, it can forms the UTP. So like that, the glycogenesis pathway, it will be occurs beyond the 6 to 8 steps. So next we learn regarding the importance enzyme. So those are involved in the regulations of glycogenesis pathway. One is the glycogen synthase. So already you know. So this enzyme is required for the additions of glucose unit to the non-reducing end by formations of the alpha 1,4 glycoside bond. And another one is the glycosyl transferase enzyme, nothing but that is the glucose branching, that is glycogen branching enzyme, that is the GBE. So this enzyme, it can transfer 7 glucose from the non-residing end to the another end. Okay, so that is the by formations of the alpha 1,6 glycosidic bond. So that is the important two. These are two enzymes. So any variations in these two enzymes, then the person was suffering from the glycogen storage disorders. So regarding the stimulation, so already you learned regarding, so this insulin is the stimulate the glycogenesis, because so this insulin, it can mainly stimulate glycogenesis by via by causing the dephosphorylations or activations of the glycogen synthase enzyme. And regarding the inhibitors, so glucagon in the liver and epinephrine in the both liver and other muscle, they can inhibit the glycogenesis by through a cyclic AMP protein kinase phosphorylations cascade by involving the deactivations of the glycogen synthase enzyme. So like that, so these three molecules are hormones. So they are mainly involved in the regulations of the glycogenesis. Insulin, it can stimulate, and the glucagon and epinephrine, so they are also hormone, so they are inhibit the glycogenesis. Okay, so regarding significance, so this glycogen pathway, it was acts as the FD or source for the energy whenever necessary, whenever the hypoglycemia or in the from the blood, the glucose level is decreases, when the body require the glucose, so on that time it can supply the, or it can initiate the breakdown of the glycogen into a glucose. So this is regarding the significance of the glycogenesis. So next one, so regarding the